Hello, my name is Daniel Gail, and my independent study was Caenorhabditis elegans, use of apoptosis in the innate immune response to Pseudomonas bacterial infections. I was advised by Dr. Strand. For many reasons, scientists have used C. elegans as the model organism of their choice, especially in studies for innate immunity. One of these reasons is that the genome is completely sequenced, allowing for shortcuts to know exactly where the gene is located. Another reason is that C. elegans reproduce rapidly due to having self-fertile females. Within a matter of days, adult worms can lay as many as 300 eggs, and within a couple more days, they will mature to adults. The innate immune system refers to a set of pathways and genes used in protecting the organism against diseases. More complex organisms combine this with an adaptive immune system that eliminates specific diseases through the use of antibodies and T cells. However, C. elegans does not have an adaptive immune system, so all immune responses will be due to the innate aspect. These tiny microscopic worms surprisingly are very similar to humans in a way. Of the 18,452 protein sequences, 83% of them have a human homologue. This helps because it's possible that the genes that we investigate in this organism can be related to similar genes or processes in humans. When introduced to a pathogen, C. elegans only has this one type of response that is based on set genes and pathways to both recognize and protect against diseases. When ingested, pathogens often create damage within a cell, which creates cellular stress and can leave behind pathogen-associated molecular patterns, or PAMPs. Certain genes can be activated in response to stress signals. The gene PMK1 is activated by phosphorylation due to wounding in the epidermis layer, while KGB1 is activated in response to heavy metal toxicity. Stress can come from anywhere, though. PK1 is activated by stress in the endoplasmic reticulum. Pathogens often create stress by exerting toxins to create a pH imbalance. PAMPs normally come in the form of toxins or other common structures typical of bacteria. The toxins create stress and can be recognized by the signal receptors. These genes are often the beginning of a much larger signaling cascade that helps mediate the defense response. These normally come in the form of MAP kinase cascades that activate through rapid phosphorylation and dephosphorylation. Cellular receptors begin the signaling cascade that ultimately upregulate defense proteins. These proteins normally come in the form of detoxifying enzymes to adjust pH balance and cellular homeostasis, or efflux pumps, to package up and send toxins to areas where pathogens are less effective. Another way that both C. elegans and humans can defend against pathogens is through the use of reactive oxygen species. The enzyme BLI3 reduces oxygen to superanions, which can be used by SKPO1 to create hypochlorous acid, which can degrade pathogens. Apoptosis has been shown to help defend against certain viral infections, and it is possible it is also used for other bacterial infections. Apoptosis is a systematic way for an organism to execute cell death. Apoptosis is often triggered by the P38 MAP kinase pathway, which includes genes like PMK1. This pathway is shown to be in higher activation in response to tissue damage. The MAP kinase pathway, among others, is a way to induce or inhibit the apoptotic pathway. The culmination of this pathway is in the SED3 gene, which is the executioner caspase that carries out the process of apoptosis by lysing cells. SED3 can be regulated by other proteins in the pathway, as shown in the picture to the side. SED3 has to be activated by SED4 protein through a cleavage of an aspartate residue. This initiator caspase can be inhibited by SED9. High levels of PMK1 from the MAP kinase pathway have been shown to inhibit SED9, allowing the pathway to continue. Apoptosis can kill the infected cells to help stop the spread of bacteria by isolating cells from the rest of the organism. A commonly investigated type of bacteria, especially in our lab, is Pseudomonas, which encompasses 140 different species. Pseudomonas aeruginosa is one of the most studied strains and is often found in moist environments. This strain has been shown to cause serious infections in both cancer and cystic fibrosis patients. There are two main ways in which bacteria infects and kills C. elegans, which are fast and slow killing. Fast killing normally takes less than 2 days and is normally the result of extracellular toxins that spread throughout the organism, triggering neuromuscular paralysis of the worm, inhibiting their ability to move and eat. Slow killing, however, takes place over the course of 2 to 3 days. Slow killing is done normally from the result of bacteria building up in the gut, which decreases the worm's ability to metabolize nutrients. My research question and goal for this study was to determine C. elegans' use of apoptosis in the innate immune response to both fast and slow killing Pseudomonas bacterial infections. My study was done by breaking up into two separate specific aims. My first aim was to classify strains of bacteria as either fast or slow killing. To do this, 3 separate strains of bacteria were cultured on 2 separate medias. The first strain was E. coli OP50, which is generally used as the food source for culturing C. elegans, while Pseudomonas 39A2 and 36C6 were the experimental strains. The experimental strains were plated on both slow kill, which is a normal nematode growth media, and fast kill, PGS media. PGS is considered fast kill because it has a higher osmolarity, which means it has a higher salt content, which has been shown to allow for more activation of fast kill genes. OP50 was cultured on NGM just so we could have a control, because we knew that C. elegans would survive perfectly on OP50 and NGM. Worms were transferred to plates and incubated with bacteria. We observed the death rates by using a touch movement assay, where we touched the head of the organism to see if it moved over a course of 48 hours to monitor the death rate. The second aim was to see how the organism used SED3 specifically, the executioner gene, throughout the infection process. At time points of 4, 8, and 12 hours in the infection process, RNA was extracted, then reverse transcribed in PCR. Ultimately, the product was visualized through gel electrophoresis to show the results. The first thing I did during the independent study process was to design and test primers. First, I tested the primers with genomic DNA and found that SED3 amplified the genomic DNA with a band of about 400 bases. The first set of ACT1 primers amplified the genomic DNA at a band of about 100 bases. However, the primer did not span separate exons, so I had to design new primers so I could use them with complementary DNA. The second gel shows the ability to distinguish between complementary DNA and genomic DNA. SED3 shows the ability to do this by showing separate bands at 400 and 100 bases, with the 100 bases being the complementary DNA from the mRNA product. The second set of ACT1 primers never showed the ability to amplify complementary DNA. Next, the infection assays were performed to classify strains 36C6 and 39A2 as either fast or slow killing. Pseudomonas 36C6 ultimately was classified as fast killing because there was an 80% increase in death in the fast killing assay, and the first death happened around the 4th hour. Pseudomonas 39A2 was classified as more of a slow killing bacteria due to increases in death, and the first death did not come till the 24th hour. This help shows that there's no true universal fast or slow killing bacteria, but a combination of both. Different pathogenesis show that they will kill C. elegans starting at different times. It was important to find a positive control for SED3. This would allow us to have a baseline for what a band should look like in a situation where apoptosis is present. hexabromocyclododecane, or HBCD, was shown in literature to induce apoptosis. C. elegans was transferred to a solution of 200 nanomolar HBCD in the presence of food, and RNA was extracted after 8 hours. This unfortunately showed no inducement of apoptosis. The last test was to see where SED3 was expressed in the infection process. Shown in the gel, there were bands of the correct size for strain 36C6 at 4 and 8 hours on both NGM and PGS plates. However, there was no expression for the strain 39A2 or for the 12-hour time point for 36C6. As a fast-killing bacteria, it makes sense that expression would occur early in the infection process, but decrease later, because the toxin could take over the body. The signaling process would recognize the toxin or damage the cell. 39A2 showed no expression since it was a slow-kill bacteria. In slow-kill bacteria, the bacteria would build up without doing much damage to create stress signals for the receptors to pick up. It would be interesting to see if these stress signals would build up at a longer time period if SED3 would be upregulated. The conclusions of the study was that Pseudomonas bacterial strains exhibit different modes of pathogenesis as seen in 36C6, which showed more of a fast kill, while 39A2 showed more of a slow killing pathogenesis. The conclusion of the second aim was that SED3 levels were elevated in the first 8 hours of the 36C6 infection. Based on literature, it makes sense that fast-killing bacteria would increase the expression levels and result in toxins. This shows that apoptosis may be used in the early immune response to fast-kill bacteria. In the future, it would be exciting to repeat the study with better controls, like a housekeeping gene with primers that worked, and a control that shows upregulation of SED3 in situations where apoptosis should occur. It would also be nice to see more strains of bacteria classified to see if the expression pattern is universal or if it's just for these 2 strains. The study would also benefit from monitoring the expression level of other genes in the signaling and apoptosis pathway in order to shine more light on the exact pathway that is taking recognition to cell death. I have a lot of people I'd like to thank for helping me along this journey. Dr. Shane and the entire lab group for being by my side the entire time and helping me whenever I needed it. Dr. West is my second reader and one of my professors that I've had more times than anyone else probably for helping me out through my 4 years. The entire BCMB and bio departments, the Copeland Foundation for funding my experiments, my parents and siblings for always being there, along with my family and rest of my friends. And I have to thank my baseball team, especially the guys at Yost House who were my best friends for 4 years. These are my sources, and thank you for listening.